In this screencast, we are going to talk about something called non-disjunction, which is related to something called aneuploidy. And all of this is related to meiosis and issues that can occur within it. So let's first take a step back and talk about ploidy. Remember that a typical body cell in a eukaryotic cell, like a human, is going to be 2N or diploid. And this is the normal condition for every body cell. Then we also have a monoploid, which is sometimes also known as haploid. And this is the condition for cells that are going to go through meiosis and that will end up as a sperm or an egg cell, right? We call those gametes. But there's a situation where sometimes you're not going to have the right number of chromosomes. So here's a normal cell, and here's our nucleus, and inside of that, we should have 46 chromosomes. That's exactly the number you want, because it means that we have 23 of those chromosomes coming from your dad, and 23 that you get from mom. But there are some situations where meiosis doesn't work correctly when we're trying to form the gametes. And sometimes instead of having an egg that has 23 chromosomes, which is what it normally has, it has something called an aneuploidy situation. Just like what we have here, where it perhaps maybe has 24 chromosomes or 22 chromosomes, or 24, or maybe less than the typical 23 that we would expect. Again, we call this aneuploidy. But usually if an egg is missing one or more chromosomes, then the egg won't be viable, meaning it won't work. But there are some situations where it could sneak through. So what leads to this situation where we have an aneuploid? Well, let's show it out. Here we have an egg with, let's say, 22 chromosomes. And then we're combining it with a sperm that has the typical 23 chromosomes. That means that we're going to create a body cell, right? The first body cell, also known as the zygote, is not going to have the typical 46 chromosomes. We only have 23 from dad and 22 from mom, meaning that we would only have a total of 45 chromosomes instead of the 46 that we would expect. Again, let's talk through a situation here. So let me get a new slide. Let's say that we have this cell, and let's make it a non-human cell and make it only have 6 chromosomes to make things easier. So there we have 6 chromosomes. If you count out the lines in there, there are 6 total. And we have both mommy and daddy chromosomes in there. We have one long daddy chromosome, one long mommy chromosome, one medium dad, one medium mom, one small dad, and one small mom. And this is a diploid cell, and there should be 3 pairs or 3 homologs that exist. So let's just number them quickly from big to little. Okay, there they all are. And we see that the 1s are the longest, and the 3s are the shortest. But I'm going to erase the numbers because they'll be in the way of what I'm trying to show you. So we know that these chromosomes first need to undergo replication, and they need to make a copy of each chromosome. There we go, and they're all sort of snuggled in next to each other, and they're going to be connected at the center by what? What's that little centerpiece? That's the centromere. And remember, this is showing the cell now after it's gone through the S phase of the cell cycle. So now here I have a bunch of homologous chromosomes in their duplicated state. But in meiosis, we always have crossing over occurring. But keep in mind, I'm not going to show it because it just complicates things too much when I'm trying to keep this on the simpler side. But I do want to show you what happens in the metaphase alignment of meiosis 1. And the chromosomes, we know that they sort of line up randomly, so let's show that. So let's say we have these, they're going to line up here, and then their homologs are these blue here. They're going to line up obviously next to each other. Then the medium ones, maybe the blue are here, and the pink are on the other side. And then for the small, oops, don't rotate, come with me, come over here, hold on, there's the pink, and then the blue. Okay, so there we go, I changed the outline a little so everything fit inside of the cell. And then of course we need our centrioles. There they are shown in black. Those are on the ends, and they're going to send out spindle fibers, which we know will attach to the kinetic cores near the centromere. Okay? And so this is a normal situation, and the spindle fibers would pull the chromosomes to the opposite poles. But let me move the spindle fibers away because they're kind of in my way of what I'm trying to show you here. So now the movement would be like that, and then these would come over here, and that would lead to those two daughter cells after meiosis 1. And then, as you can imagine, this is normal, and the sister chromatids would split in half, right? They would split into 4 daughter cells after meiosis 2 occurs. But let's go backwards and reestablish our spindle fibers. But imagine in this situation, this works properly and this works properly. That side's fine, but here it only attaches to 2. And instead, from the east side, we have this one pulling it across to the other side. So that extra spindle fiber will attach to the kinetochore of that pink chromosome here, where usually it would be coming from this right side. And as you can imagine, this is going to create big problems when the cells pull apart. So let me show you what that would look like. We would get this, 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 and this, all in this cell here, like shown below. And then in this cell, we'd only get these long blue and these short blue chromosomes. And this is only after meiosis 1, right? So let's move things up a little. So from this cell, when it splits, we're going to get 1 big pink, 1 medium pink, 1 little pink, and 1 medium blue. And this other cell will get the same. They'll be anywhere in the cell, so even though they don't look identical here and here, their chromosomes are the same. And then in this cell, we have even more problems, right? We're going to get 1 tall blue and 1 little blue. Let's draw that in. 1 big, 1 little, 1 big, 1 little. Now the problem is that we're expecting to have 3 in each of our resulting daughter cells. But in this one, we have 1, 2, 3, 4 in this one, 2 in this, and 2 in this. So all of those situations are not good, not what we want. The gametes here, undo, over here, they have an extra chromosome, and over here, they're lacking a chromosome. So none of these have the correct number of chromosomes. All will be what we call aneuploids. And it leads to serious issues if you don't have the correct number of chromosomes, which you will or have already learned about when you watched a screencast on syndromes, right? So keep in mind that there was this event where the spindle fibers did not break apart correctly. And this was again in the first meiotic event, shown here, where the spindle fibers again are not connecting in the right spot. And that process, when things don't break apart correctly, has a name. We call that non-disjunction. And non-disjunction leads to an aneuploidy situation. And remember, aneuploidy is when you have the wrong number of chromosomes in a cell. And in the example we just talked about here, this was happening again in meiosis 1, but what would happen if it happened later on and was happening instead in meiosis 2? So let's go back to an original cell situation. We have long, medium, and little pink and blue chromosomes. Now let's line the chromosomes up in metaphase. Now I may have organized these different than I did in the last one, but again, here we are in metaphase 1. They line up properly. The spindle fibers connect properly, and these are our resulting daughter cells. There are 2 of them, and they have the proper number of chromosomes. But now imagine there is a non-disjunction event that happens in the second division, and we don't attach the spindle fiber to the sister chromatid associated with one of these little chromosomes, let's say. First, let's move these where they need to be. Okay. Spindle fibers. Attach, attach, attach. Fine there. Let's move these around. Up. Down. Oop. Down. And here we have spindle fiber that attaches here and here. Sorry, I forgot to show the other half here. So add these spindle fibers in. They're fine. We have these 2 lining up. We're going to say that by accident it forgets to attach here, but it'll be fine on the other side. Okay, now what does that look like? Big blue chromosome, medium blue, small pink. We're getting the same in both. Shown there. Those are fine. We're expecting to have 3 chromosomes within here and here. Now here's where the problem lays. We will get 2 chromosomes in this cell, but spindle fiber forgot to attach here. This cell should be fine because it attaches to the 3. So in this cell, we'll get this big pink and little pink. I think that was medium actually. And then in this cell, we'll get the little pink it attaches to, big pink. We'll also get the medium pink there. And then this spindle fiber attached to both of these, so we will get 2 of the little blues. The entire thing will go over that way. So we know that in situation A and B we're fine. We're expecting to have those 3. But in C and D, we do not have the correct number. Here we have only 2, and here we have 4. So both of those are aneuploids. So remember in the previous condition that we talked about here, when non-disjunction happens in meiosis 1, all of the cells are not viable. They're all aneuploids. And problems like that can be pretty catastrophic. It can wipe out an organism before it's born. But in this situation, we have 2 viable cells and only 2 are aneuploids. Some syndromes, as we know, do result, and remember that syndromes are the result of something called aneuploidy, when you have the wrong number of chromosomes in a cell, and the gametes are coming together with abnormal numbers of chromosomes. And Down syndrome is the one that you're probably most familiar with. That ends what I wanted to tell you guys here. So any questions, let me know.